After the discovery of large number of type 2 restriction endonucleases, then scientists proposed that there must be some sort of naming mechanism, which we can call it as the nomenclature of restriction endonucleases. So one of the first proposal was given by Smith and Nathans in 1973. So now we are going to discuss the key features of this nomenclature of restriction enzymes. So one of the first point was that the host organism that produce this restriction enzymes, the name can be after the name of that organism. For example, if the restriction enzyme, it is isolated from Escherichia coli, so the first word will be from the genus name, that is Escherichia. So E will be taken from the first name, that is the genus name. And the next two words of the restriction enzyme will be selected from the specific epithet, that is the second name of the species, like coli. So the CO, first two words will be selected from specific epithet. So the name will become ECO. ECO means that the restriction enzyme that has been isolated from Escherichia coli. In the same way, the restriction enzymes that were isolated from Haemophilus influenzae, they were called as HIN. It means that capital H from the genus name Haemophilus and IN from influenzae, first two letters of influenzae. Then in the second step, the name of the strain can also be given. Now, the single species of microbe, it may be represented by different type of strains that may be isolated from different ecological sites. तो जो strain का नाम है उसको species name के बाद include किया जाता है. Like E. coli का जो strain K है इसको लिखा जाता है like ECO K. And in case of Haemophilus influenzae, the strain D, it has been reported to produce different type of restriction enzymes. Now, there may be possibility that strain D, that now called as HINT, it may produce more than one type of restriction enzymes. And if this is the situation, तो फिर HINT के बाद number of restriction enzymes लिखा जाता है Roman numerals में, like 1, 2, 3, HINT 1, HINT 2, HINT 3. As I told you that type 2 restriction enzymes, they may have separate enzymes for restriction and modification. So they may also be differentiated कि क्या ये restriction वाला enzyme है या ये modification वाला enzyme है. तो पूरा नाम लिखने के बाद like HINT 3, तो उससे पहले लिखा जाता है अगर तो ये restriction enzyme है तो letter R is added like R dot HINT 3. It will indicate that this enzyme, it is showing restriction activity, and if it is written as M dot HINT 3, so it means that this enzyme, it is showing methylation activity and playing some sort of role in DNA modification. So these are examples of restriction endonucleases nomenclature, that how they can be given different type of names. So if we take the first example, like the SMA1, SMA1. So actually this enzyme was isolated from Serratia marcescens. So S is from genus name and MA from specific epithet, and it was the first enzyme that was isolated. So SMA1 name was given, and this is the recognition sequence, and it is a 6 base pair recognition sequence CC triple C GGG. And then another example, it is the most common one that we are using, it is HINT 2. As I told you that it is isolated from Haemophilus influenzae. So HN from the genus and specific epithet, and D, it is from the strain D, and it is a second enzyme that was actually isolated. So the name is Hind2. And from the same strain, strain D, the third enzyme was also isolated. So the name Hind3 it is given here. Then another important enzyme, it is the BAMH1. So it is actually the BAMH1. So the B, it is from Bacillus, and AM from amyloleucofaciens, and the strain, it is H, and it is the first enzyme that was isolated. So this enzyme is BAMH1, and this is the recognition sequence that was recognized, and it is a 6 base pair sequence like GGATCC. Another example is the recognition sequence that was recognized by Hind2, and it is represented by G, then T, then any purine base can be there, pyrimidine base, and then any purine base it is there, then adenine and cytosine. So the enzymes, they may have different restriction sites that we are going to discuss in next chapter.